Thanks so much for joining up with ESS-DIVE's April webinar, our monthly webinar series. My name is Rob, and I'm a postdoc with ESS-DIVE. First, thanks everyone for joining, and I just want to do a quick introduction to the topic of our webinar this month. And yeah, just a very brief introduction to our presenter, who we are so thankful to have joining us for the webinar. So Pamela Weisenhorn from Argonne National Lab is going to be presenting about the progress on a community reporting format related to microbiome data. So this is Amplicon sequencing data, and demonstrate some use cases for how these sort of standardized data formats can help link both the data in ESS-DIVE and the data that's in KBase. So with that, I'll turn things over to Pamela. And again, thanks for being with us for this webinar, Pamela.
Thanks for the introduction, Rob. So if I can share my screen here. Okay. Does that look good to everyone?
Looks great here.
Okay. So this 16S Amplicon reporting format that I've been working on with ESS-DIVE is one that I've developed in coordination with KBase, so where I am the product and science lead for Amplicon data, input the data type itself, as well as the analysis and processing pipelines within KBase. And so being in this joint position really gave me the ability to make sure that there's sort of a seamless transfer between data. And so I want to talk a little bit about that and the potential for that with not just the Amplicon, but also samples and other reporting formats as sort of using the standardization to allow data transfer. So I'm going to start off by giving some details on the reporting format. At this stage, this is not a finalized format. I'm actively seeking community feedback and looking to modify this. So all of the pieces that I'm going to be talking about here today are still sort of in flux and will be until we get the reporting format finalized. But we've been able to make a lot of development within KBase based on sort of the format as it's described here. And so I'll talk a little bit about how we're using that format to in KBase to create a data type, as well as how it's integrated with the sample reporting format to support our analyses and the potential for integration with other data types in KBase. So first, to talk a little bit about the reporting format itself. It's a multiple file reporting format. So there's a number of different data types that are unique that need to be saved. And so here there's sort of the Amplicon or taxon table itself, which has sort of the raw counts of how many of this organism are in this sample. The consensus sequence, which is actually the assertion of taxonomic identity and is most closely linked to the actual raw data. So everything in the taxon table is an assertion, but we want to keep that linked as closely as possible to the data themselves. The key metadata, which describe how we went from the consensus sequences to that table, as well as sort of an optional taxonomy file, which is just another level of assertion, which would assign a specific name to each of the organisms within the taxon table. So going through each of the files, and please don't hesitate to interrupt me or call attention to things. I don't see the chat here in my window, so if somebody will stop me if someone has a question, that would be great. But So talking about the main data is the taxon table. It's a tab delimited text file. This is one of multiple standards that are currently adopted sort of broadly through the community of researchers who are using Amplicon data. A couple other common formats would be a biome or biome 2 file. They're all very similar in terms of their formatting and the data that they hold. Biome and biome 2 compress the data a little bit more because these are count data. There's a whole lot of zeros here. It's a sparse table. So biome and biome 2 take advantage of that to make the file size a little bit smaller. And they also have a different way of storing taxonomy information, but for now, we've decided to go with the tab delimited text file because that's more accessible to researchers who aren't very hyper familiar with Amplicon data itself. And so for incorporating this data potentially into other models and systems and questions outside of the context of strictly microbiome studies, I wanted something that was a little bit more accessible to the community. So here we have a really simple sort of Excel spreadsheet type file where the, and I have this drawn wrong, rows should be sample IDs here, and then the columns should be taxon IDs. And so that standardization just allows the input into KBase to go more smoothly. And then this table itself holds raw count data, and it's important that it holds the raw count data. That's usually not what's used in doing the analysis itself. But because the metadata file that we talk about later doesn't capture any of that post-processing, and that post-processing is available within KBase, which is a system that captures provenance and all of the different steps that are taken in a reproducible and shareable way. We have gone with the raw count data as being what's stored here because all of the other data can be derived from it. So if you wanted to do a proportional abundance or absolute abundance, which requires the input of additional data, that can all be reconstructed from the raw count data. So for the standard reporting format, it's raw count data. So next is the consensus sequence file. The names must match the taxon IDs in the taxon abundance table. So this is the link between the raw sequence data and the taxon assertions in the taxon abundance table. And so it's critical that the IDs be clearly and easy to link. So it's got to be an exact match. There needs to be one sequence per taxon ID. And this is because these sequences are how you derive the taxonomic identity. And so if you had multiple sequences for a taxon ID, so you clustered them, but then you capped each of the individual sequences, you might actually get different taxonomic assertions. That's really common with these data. And so you need to have one single consensus sequence per taxon ID. This link, this ID serves as that link, again, between the taxonomic identity and the raw sequence data. One reason for having this file is also that ESS-DIVE is a data repository, but it doesn't hope to archive all Amplicon data ever. There are other existing repositories for the raw sequence data, and so this would only be holding sort of this process data on which assertions were made. And it's stored in a relatively common FASTA file format. So this is something that anyone who's processing these data should be relatively familiar with. Yeah. Moving on to the required metadata, and this is where things get perhaps a little bit more questionable in terms of what could be included or not.
Hey, Pamela, can I, sorry
Yes,
to interrupt you.
please.
Can I just ask you something still regarding the previous slide?
Yes.
So those representative sequences, then this would be, could be either ASVs or OTU representatives. Would that be it?
Yes.
Okay.
Yes. So we are agnostic as far as whether these are clustered or how they're clustered. We'll get to that in the metadata, how you specify that. But it's important that there's just the one sequence per ID.
Okay, and the FASTA file would then be, sorry, just the representatives. So meaning that the raw data behind this is not deposited in ESS-DIVE. We have the representatives and we have the mapping files.
Right.
Okay, got it.
Right. So the name here would serve as a mapping between the representative sequence and the table itself. So that needs to be a match. So you don't actually have to have a independent mapping file. But if you're talking about mapping in terms of metadata, we'll actually get to that later. That's
no,
here,
I mean
that's
just
covered by
sequences,
sample.
right? Okay.
Yeah.
So here it's essentially all the sort of process data that anyone could just download and to reproduce the analysis, essentially.
Yes, yes, that's the
Okay,
thought.
got
And
it.
then the raw sequence data would then be deposited in like NCBI or
Yeah,
somewhere else.
SRA, yeah. Okay.
Yeah, yeah,
Perfect, thank you.
yeah. So the ESS-DIVE isn't storing something that's stored there as well. So for the required metadata, there's both sequencing and processing metadata. Here, I really tried to strike a balance between the two hats I was wearing with ESS-DIVE and KBase. So with ESS-DIVE, we wanted to have flexibility in our reporting formats. We didn't want the bar to be so high that it made it very difficult to store your data. We want people to actively be using ESS-DIVE to archive and store their data and make it accessible and findable for people. On the other hand, in KBase, we try very hard to enforce reproducibility and to really track what has happened to data, how data has been processed. And on system within KBase, we do that very tightly. But here, this processing is happening off system. And so on your own computer. And so we're trying to capture sort of the critical metadata. And from studies I've read, I've chosen what I think is the most critical, but I think that's really open to debate. And I'm hoping through the surveys that I'm working on now that it will be sort of broadly dispersed to the community to get feedback on that, as well as some feedback on whether this is too high a hurdle to require these things, or whether the community would like to see more required in order to really increase reproducibility of the data. Because I think with Amplicon data, there's a lot out there, but there's also a really big challenge in reusing that data because a lot of these metadata on the processing are missing or incomplete. But for this reporting format, sort of the happy medium I've found for the time being is for sequencing to require the target gene. So is this 16S, 18S? Is it a functional gene? The target region of that gene. So is it the full sequence? Is it a V4 region? The forward and reverse primers that were used. So that's even a little bit more specific than the region on the gene, as well as the sequencing platform that was used. So is this PacBio data? Is this Illumina data? Because those each have very different error structures as well as read lengths. And so it's important to try to capture that and not cross those data streams. And then as far as the processing, there's a quality filtering cutoff, as well as the clustering cutoff. And so that kind of gets a little bit to your question earlier, Ricardo, of OTU versus ASV, but it's sort of a little bit more broad. So this is what's required.
Can I ask you something?
Yeah.
Sorry. So also regarding the clustering, okay, I think you're getting to that, like clustering methods and denoising. I was actually going to ask more specifically about chimera removal.
Okay, yeah. So
if it has been performed and how and so on. Just beyond the cutoffs, I think that's really fundamental. I would suggest. I struggled so much over the years with that, with bad data out because of chimeric sequences, that I'm just really kind of really set on it.
Yeah, so chimera removal method and whether or not that process had happened was definitely on my list of my initial brainstorming list of all of the different things that you might want to include. And so I'm happy to hear you say it, and I hope that as I get feedback from the community, I'll see if there's a consensus on that being important, because I agree. I think that that's as important as the quality filtering cutoff in terms of removing data that may not be particularly good.
Yeah, right. Yeah, exactly. Because it's just, you know, I'm just really kind of really want sort of to make an argument for this, because especially when you're dealing with really large data sets. I mean, if we're kind of, let's say someone gets the data out and wants to make some whatever phylogenetic assignment, reconstruction, if you're dealing, when you kind of working with Sanger sequences, very few sequences, one can sort of see this visually during the processes. But with this amount of data, it completely distorts the results, and it's just impossible to handpick these sequences. It's just, you know, it can be really months of work thrown away if one realizes that the data was full of chimeric sequences. So just knowing if it was done, it's just, I think, such a fundamental point.
Yeah, I am certainly happy to add that to the list of things, and I was hoping to get that kind of feedback today before I send out the survey. For, you know, I'm going to have in that survey, I'm going to have, this is what is included, this is a list of things that aren't included but that people have suggested should be included, and kind of get the broader community's feedback on that. So some of the early feedback that I got was that I was asking for too many things and being a little bit too detailed. And so that made it too hard for people to go through the process of generating this file that contained all the information. But I'm hoping that we can find ways as a community to better track the methods that we're using and how all of this processing is happening.
Yeah, definitely sounds good.
So some of the optional or omitted metadata would be sort of the extraction kit or protocol, sequencing run, the sequencing kit in chemistry, as well as the sequencing center. All of these things have been shown to cause variation in data sets. If you take the same sample and you send it to multiple places, you get back different communities. As well as for the processing, the clustering method and then the denoising method are both very important, and they're currently included as optional. But the sequence depth cutoff for samples, so knowing whether other things were thrown out that may not have been thrown out in a different data set. Barcode error threshold, so how sure are you that it's really from that that particular sample. And then just sort of the reminder that the data in the table should be the raw count data, because certainly there are a lot of downstream steps from here in terms of normalizing that data or making it into relative abundance data or proportional data, and all of those are critical as well. And so we stop here at the raw count so that those steps could ideally all be done after any sort of combination of data or comparisons of data are made. So I'm going to pause just for a second to see if anybody else has thoughts on other data that they might think should be included, or arguments for why any of these things should be thrown out. Okay, hearing none, I'll start moving forward. And again, I'm going to be sending out a survey, so I hope to get a better and broader feel from that on exactly what should be included or excluded here. And now there's sort of a fourth file, and this would be an optional file, and that's one with taxonomy. So with a name assignment for each of the taxa in the taxon table. So this is another area where there's a lot of variation. And so I have it stored actually as a separate file in and of itself. But for that file, there would need to be sort of that the taxon ID with it linked to the taxonomic string. So this is the assertion of the name, the database that came from, the version of that database, and also the assignment algorithm. So each one of these steps can be highly variable, and depending on the algorithm used, in some cases, the order in which the data are read into these tools to assign taxonomy, you can get different names out. And so in the interests of reproducibility with the taxonomy, this would be stored as a separate file. So it'd be available to people who are interested in looking for specific taxa or in pursuing that, but maybe don't know how to do these assignments themselves. So somebody who's maybe a little bit less familiar with microbiome data, they would have names available to them that they could start to work from. But it would also provide enough information to make sure that the data are comparable across data sets again. So this is a little different than how it's stored a lot of times now. Again, the biome file would contain both the taxonomy data as well, but in the interests of broadening out beyond just the community who's actively making the data to people who might be interested in using the data and not creating it, we have a separate file for that has a direct key ID link with the taxonomic ID. So now I'm going to switch gears a little bit and talk about how we leverage this reporting format to get data into KBase, and then we can process that data within that system, and some of the advantages of sort of generally of having these reporting formats and having this level of standardization. And sort of how that would help move analyses forward, especially sort of cross-study analyses within the DOE system. So the data upload process, you'll see that the target gene region, the primer sequences, the sequencing platform, the quality filtering cutoff, the clustering cutoff, and method. So the metadata file basically needs to be manually re-entered right now into our uploader. And I just wanted to acknowledge Tian for his work actually creating this uploader within KBase. And we have some validation of the field contents during the upload to KBase. So for example, the primer sequence has to be a string. You can't throw numbers in there. And this will sort of be solidified once the reporting format itself is finalized. And so this is sort of a little bit of that iterative development between the reporting format and the tools that are in KBase that's been going on sort of behind the scenes. But for now, what we have is sort of a manual re-entry of this, this uploading process.
Pamela, can I just ask something? So here in the primer sequences, where is the orientation of the primer specified?
If you have sort of the hover over, it specifies the orientation.
Okay, perfect.
But
perfect.
since it's not live, you can't. Yeah.
Okay. So you can just
yeah.
over and you see whether it's like a 5' to 3'. Okay.
Right. But we don't have that here. So yeah. So we have the sort of final decision on that validation once we know exactly what the fields are. But that also, once we have that finalized, has sort of the potential for doing an automated parsing of the metadata file. So right now you have to manually re-enter things, which at least when I'm doing it, involves a lot of copying and pasting of my primer sequences. And so that that has a large opportunity for error or mistyping. And so if this data is already recorded in a metadata file, there's potential to go through and do that parsing. And so I was curious to ask people here whether that would be a useful feature for you. And maybe we could do sort of a show of hands if people want to raise their hand, and maybe Rob or Joan could sort of get a feel for if anybody is interested in seeing that develop. So that's sort of a easier thing for us to do. Again, once that's final validations, files, everything are created. Another potential future capability would be to have sort of a KBase suggested package contents. And so ESS-DIVE, again, in the interest of that broad flexibility and wide adoption, doesn't specify what is contained within a package. But KBase could certainly have sort of a suggestion on what a package could contain. And namely, it would contain the 4 files that I just mentioned, as well as the samples file, which would contain information on the sample itself. So sample metadata, like the location where something was pulled in, the biome it's from, and then perhaps recommend also sort of a predictable file name. And then that could enable sort of seamless upload of an entire package from ESS-DIVE for the KBase to go through and load data into the correct data type. So right now you have to, in the upload process, you have to specify your taxon table, you have to specify your consensus file, you have to manually enter the metadata, you can specify sort of a taxonomy, and then samples are actually uploaded entirely separately and on their own. So I wondered then if sort of a similar raising of hands, if users here would take advantage of that feature, or is it not outweigh the advantages of having the greater flexibility of the package contents and file naming?
Would this then impose exactly what would go in? Or I'm just wondering how compatible it would be to have such an automated system, but kind of like with minimal requirements, but still retaining the flexibility for anything else that we would like to add.
Right.
Does that make
so
sense?
like if you have additional files that you wanted, but that would have some name that didn't fit into this predictable namespace. So like you want to have a file on soil chemistry or something that's separate.
Yeah, for example, yes.
Yeah.
But I think like the automation, especially from an ESS-DIVE prepackaged bundle, I think that would be really useful.
Yeah, I think it could be. Yeah, I think it would be possible to sort of ignore files, or maybe have a sort of interactive specification of which file goes to which of these table types. It reduces the automation, so it puts more onus on you to do that at upload, but it's still relatively, it's still easier than running separate apps to get this data into these data types. So
yeah, it makes sense. I think, yeah, having kind of like a core, say, files, formats, and whatever, something that goes automatic, something that could be automated. Let's say you have a ESS-DIVE package with a bunch of stuff, and KBase would sort of import these core minimal, say, files requirements, and then having some sort of interface where you could sort of complement this in a semi-automated way. Does this make sense?
Okay. Yeah. I think that that makes sense. As you'd say that maybe something in between those two options would sort of be the ideal place in your mind for that, where that trade-off is between flexibility and we'll call it convenience of upload.
Yeah, I would say so. I think that, like, for convenience, we not always have, you know, the immediate time to just kind of go through the whole process, but just having something that's automated and puts there the minimum, like the essential part, and then still leave us the flexibility to, you know, complement it afterwards, or if we just have more time, or if you might just not even be interested in putting anything else, but just having the easy, the fun, the let's say the essential part easy and automated, and then the extras on top, let's say, in a more flexible way.
Yeah, that certainly makes sense. I think that's a really good suggestion. Does anyone else have thoughts they wanted to share out loud? Okay. So now I'm going to talk a little bit about samples. So in the interest of kind of going through this from the KBase perspective, I need to kind of back up a little bit and talk about sample and how sample's been used in KBase. You know, I want to acknowledge here Joan and all of her work in coming up with sample, and the KBase team has definitely leveraged that. So, you know, Joan's reporting format suggestion with sample is to use the CESAR and IGSN system. And so KBase has adopted that format as one of the data formats that are supported for sample upload. So within KBase, it's not restricted to the CESAR format, but that is one of the file format options that can be uploaded into KBase. And these samples then we've done a fair amount of work, and some of it is based off of Joan's crosswalks between CESAR and other sample data, metadata file formats, and such as MixS. So we have a validation of fields and then an integration with KBase ontologies. And so what that means is when you're uploading these data, if you have the latitude and longitude of samples, we'll make KBase goes through and validates and makes sure that those latitudes and longitudes make sense. The same goes for any of an array of other fields, and ensures that, you know, everything looks like real data. Obviously, there are lots of places that can fall through the cracks, and this is a much harder task than getting data uploaded and into a data type. Actually going through and doing careful validation with good error reporting is always a challenge, and it's something that's still in active development, but we're hoping to release samples as well as this Amplicon stuff in the near future. So we're getting close and being able to release this sort of broadly to the public. And if there are anyone who's interested in doing some beta testing on these for us, please also reach out. I would be happy to put you in touch with folks at KBase who are looking for people who have these data types, who are interested in using KBase to process them, so that we can get feedback on the process, and especially sort of where the hurdles are in getting data in, and whether, you know, our validators are kicking out data that should be accepted, or letting in data that shouldn't be. But here, there's also sort of the potential for future development. So one of our sort of things that's in earlier active development and testing would be the ability to upload samples directly based on the IGSN after those samples are made public. So instead of having to upload the file itself, you can just specify a list of IGSN numbers that are comma separated, and then it'll pull straight from the GeoSample system and pull that data into KBase. So it takes advantage of the validation that occurs there, as well as the publicly available data APIs that they have. And again, Tian has been the one who's primarily been working on this, and I didn't have a picture, so I forgot to mention, but I do want to say that Sebastian Labra has been leading the sample uploader development with a lot of help from a good number of people on the samples team in terms of getting that functionality in the system. And so, you know, it's right now the GeoSample system is easily accessible. So it's possible to just specify an IGSN and get samples uploaded. But we were wondering if people would take advantage of a similar feature with sample upload from ESS-DIVE of public samples. Right now, it's not currently possible for us to get direct access to these public samples, but it's kind of out of curiosity, would others be interested in being able to do that? So specifying like a package name, or otherwise linking in with ESS-DIVE. I'm not going to try to pigeonhole anybody into exactly how that would look at this time, because this would be sort of far future development, but interested in seeing or hearing more about that. Okay. And so then I want to talk just a little bit more about the integration with other data reporting formats and user facilities. So I'll say within KBase, samples is how you can link data that are associated with the same study, which allows us to employ lots of our downstream analyses. So for example, if you brought Amplicon data into KBase and you are interested in knowing sort of which samples were different from each other, or whether there's statistical significance, you could run a permanova or an analysis of similarity. You could make PCA or NMDS ordinations of that data, as well as doing hierarchical clustering. We have a lot of these sort of downstream analysis apps that are available. But all of those are based on looking for differences in groups of samples, or with data that is outside of that taxonomic data that's in the reporting format for 16S itself. And so that's where this sort of use of samples as an integrator within the KBase system is really critical. And so you know, I want to think a little bit more broadly sort of about that overlap between data types that are within KBase and other data formats that are being produced broadly within the DOE community. And so Joan, who led the work on the sample ID, is leading a working group right now to sort of advance using sample IDs to link data and samples across user facilities. But I also want to make a sort of parallel push here for within the KBase system, we use sample ID to link different data. So if you have, say, geochemical data from soil or water that has the same ID as the Amplicon data, you'd be able to pull both of those different matrices in together to do some of our analyses. So you might want to do a Mantel test to see if the geochemistry is explaining community composition, or a SIMPR analysis or something along those lines. And so right now, some of the data types that we're actively working on in KBase, so it would be a really great time, I think, to start to pull together and really actively pursue these linkages, as well as finalizing both the reporting formats that people are using along with the data type development in KBase. So there is still sort of this this very close relationship that would allow either seamless transfer or easy tools or scripts that can be used to convert data in an ESS-DIVE reported format to a data type within KBase. Again, with the recognition that because they serve different purposes and slightly different communities as well, ESS-DIVE has, in general, more flexible reporting formats that then would need to be standardized in a harder way to match up with data types in KBase, which has much stricter requirements because they're doing analysis on the data themselves. And so those data types need to be quite well defined. And so having sort of some understanding of those things. And so sort of the ones that come to mind and the user facilities that come to mind include metabolomics data that might be generated from MSL on the same sample that you may be measured in the field or lab, other geochemical data on extracted sequencing data of some sort. So Amplicon or metagenomics or metatranscriptomics data. And you want to pull all of that data together in an analysis. In order to be able to do that, you first need to be able to link the data. And so we need to get user facilities using the same sort of sample IDs. But then also you need to be able to get that data into the same place to do that analysis. If you want to do that analysis sort of in a broad way, right? So if you want to do it with your own sample, maybe you write a script and you're happy. But if you want to do some of these cross-comparative studies, it would be easier to pull in more standardized data. And so this is sort of open question of how people are thinking that they might be wanting to use the data, and whether that standardization is necessary. But I think it's a conversation that the community should be having. And so this is sort of my advertisement slide for these conversations and for reaching out to people to start and talk on this. And I think now is really the time to make it happen. There's a lot of work being done at a lot of different user facilities to sort of develop tools for these analyses. And so I think it's a good time for us to be working together as a community to pull these things together in a clean and efficient way that I think could save everybody a lot of energy. So that's my presentation, and I'm open for any questions or other feedback, or maybe even just broad discussion of how we can start linking data together.
Thank you, Pam. This is Charu. I had one question regarding your last slide. And, we do have a reporting format that ESS-DIVE is working on for geochemical data. Specifically, that's Kristen's work. And she is also using IGSNs for the samples. And I was curious whether, I don't know how strong your data typing is for geochemistry within KBase, but whether we should be having a conversation on importing geochemical data from us, because that's more likely, a lot of data will be stored on ESS-DIVE related to sample geochemistry.
Yeah, I think that we absolutely should be having that conversation. Like I said, that data type is not quite as far along as the Amplicon data type. So that was developed afterwards and is still very much in active development. And so as I know that that reporting format is also in active development. So I think it would be really good for us to be having those conversations to make sure that transfer could happen sort of in some of the ways that I've proposed for the Amplicon data as well. Because yeah, I think you're right. I think that there's going to be a lot of that kind of data, as well as a lot of data, from being generated by IMSL and JGI that could be pulled into KBase for the analyses. So
yeah, and it's also really exciting to see all the work you guys have done using the sample standard. And I guess I just wanted to follow up because I'm curious what kind of validators you're using on the samples, because that's something we want to also eventually incorporate the ESS-DIVE ourselves. So it might be, interesting for us to just have a follow-up conversation with your dev team to just sit down and see how you guys are doing it.
Yeah, like I said, so we've put, there's been actually quite a lot of effort that has gone into developing validation for individual fields and deciding which fields are going to be validated versus, sort of the user specified. We hope it's not too bad, with the intent of also allowing users to specify their own ontologies if it's a field that isn't in our system, but they are using a publicly available ontology. And so I would say Shane Cannon's probably the person to reach out to on that, because he's leading the folks who are developing it. Sebastian's done most of the Sebastian and Jerry have done most of the actual work, but and Shane also works across with NMDC as well, so sort of broadly. But yeah, no, I think that there's probably a lot of work on those validators that could be shared in the same way that we are definitely leveraging all of the work that Joan did in her crosswalk in terms of looking at our sample stuff. So I think there's room for KBase and ESS-DIVE to share labor on that.
Hey, just want to ask you, so what's the timeline or the status of the implementation of this integration into KBase? So let's say, for instance, I got a bunch of 16S data that have actually already been published or are in the process of being published, in a publication, and they have all IGSNs, they have, say, ESS-DIVE package. So what would be now the next step? How far can I already bring this to KBase?
Yeah, so right now, it's pretty early. So what you can do right now would be, okay, I have these files and I can upload them into KBase, sort of specifying each of the files manually, as well as for that metadata, again, having to sort of manually go through. The timeline on getting this developed, I need to get my reporting format done soon. So within probably the next quarter, I hope to have that with enough feedback to finalize that reporting format. Once that happens, it can be pretty quickly adapted in terms of parsing the file, because we've been working on the validation steps sort of all the way through. So I would say probably within the next 2 quarters, we would have something where we might be able to go through and parse that metadata so you don't have to manually enter it. As far as being able to specify package in ESS-DIVE, I would say that that's sort of a long-term future. Right now, there isn't that direct exchange. I'm proposing here that that would be a really useful and valuable thing, I think, for a lot of people. But ESS-DIVE and KBase need to discuss how to get that data back and forth. Whether it's sort of a Globus transfer, do we want publicly available data through an API? Do we want KBase to have sort of a more protected way to get data so that, you know, the publicly available data is available to KBase, but maybe not to everybody through an open API? Or, you know, what that process would look like isn't specified yet. So that's sort of a long-term future goal.
I could just add something quickly on what we want to do with this working group across the different BER data systems. So we're currently just getting started, and we're hoping to use your project, Ricardo. We've talked about this a little bit, but just starting to put in links to related data and resources across the different systems. And right now that's going to be a manual process, so we can just figure out like exactly like how to use the identifiers and how to link those to other related identifiers in a standard way to do that. So it's kind of all of this is in progress right now. So if you're interested, it's a good time to like provide feedback or get involved on what kind of tools that you might be interested in or would help in your work.
Okay, so for instance, would it be, let's say, talking about this example that I gave earlier, let's say that I kind of get this data into ESS-DIVE to begin with. So that's kind of what you have been discussing already, just to get the packages and so on. And would it make sense then, for instance, to try to work with folks at KBase to try to test the IGSN? I don't know how far they are with this. I'm just kind of trying sort of to sense a little bit what could be tried out. So even if it's not implemented, but say that I have IGSNs already, and if this could be a good time to reach out and try to test this out, for instance, independently of how then it gets connected to ESS-DIVE, but just sort of trying to get data both places.
Yeah, so for the KBase side of things, like I said, we have in development tool that you could get access to for like listing out IGSNs and pulling in the sample data. So that could be tested like right now. It may or may not work. You know, it's in active development, but it does exist and could be tested. So it would be a good time to get feedback from people who have sort of that. Again, as long as it's public data. We can't get private IGSN data, so it has to be released. But did you have anything to add, Joan? Or
Not much to add other than we are starting this use case work where we're working on connecting related samples, and so I'm hoping to work with you more on that, Ricardo.
Yeah, sounds good. Sounds pretty exciting, actually. It's like hearing how things are evolving on both hands, and it's yeah, I just kind of was sort of tasked with taking care of this data deposition, not only microbial data, but also biogeochemical data for our TES SFA at LBL. So I have sort of a lot in my hands, and I guess that I will start with the molecular data stuff that has been published or is already deposited, because that's also the type of data that I'm more familiar with. So it would be really good to kind of try to connect this from the beginning, you know, just trying to, yeah. Pretty exciting. Thanks a lot. It's great work.
Pam, one comment regarding getting data from ESS-DIVE. I think Val can chip in. But there is sort of a future milestone, I think, towards the end of this year or early next year about making our data public through our public data available through an API. And you could then query the DOI and retrieve the data. So that might be sort of down the lane when you're thinking about linking, that might help.
That's good to know.
Val, you have anything to add on that?
No, I think the only thing that I would say is that anybody who is a registered user of ESS-DIVE has access to their own data sets to download. But what we'll be doing is, so if you want to know what the API is and learn it before it becomes generally publicly available for anonymous search across all of the corpus of data, you can always go to api.essdive.lbl.gov to see what that API looks like and play around with it until then.
Okay, great. That's helpful too. I mean, I know that we had reached out to see if we could get sort of some public data to test things for development. And because we're worried, obviously implementing tokens within KBase to try to log in as a specific user is takes a lot of work on our end for the development side of things. But yeah, we could play around with it separate from KBase to get a feel for it first. That's a good idea.
I guess, Pam, sort of as a last question, what's the timeline that you have in mind for wanting to finalize these reporting formats and publishing them? Because that might help sort of give a bounding box on how much feedback and what's the timeline for people to give you feedback.
Yeah, so I'm hoping to have by the end of this fiscal year get a lot of this stuff wrapped up and out. I know that we're planning on doing an initial release for KBase of this of the Amplicon pipeline and everything before then. But that doesn't mean that it has to be completely finalized before then. So the surveys are going to go out shortly, and then probably within 2 to 3 months after that, I'm hoping to have that survey data back and be going through that, and I'll be trying to during that same time period reach out to people who are interested, or hopefully hearing from people who are interested and have strong feelings about it to discuss so that we can really get it finalized. But yeah. Fiscal year is when I hope to have a finalized reporting format, or at least to the point where it would be minor changes if anything needs to adapt in the future. I know that we want to have some adaptability and flexibility in those, but.
Great. Well, we're right at the top of the hour, so first want to give a big thanks to Pamela for sharing all the work you've been doing. And I think there were some emails in the chat for follow-up and participating in the survey that they're going to send out. So yeah, thanks so much, Pamela, for sharing. And we'll be posting the recording of the webinar too if folks here want to distribute it as well to their communities. Yeah, so we'll be posting that on our website within a day or two. So thanks, Pamela, and thanks everyone for attending.
Thank you all.
Thanks, everyone.